Hello everyone, welcome to our YouTube channel. Today we are going to talk about the basics of DNA replication. Now, before starting to know the replication of DNA, we first need to know how the information that is stored in the DNA, that is the genetic information, how it flows from one molecule to another molecule. Now, this flow of genetic information was proposed by Francis Crick in the year 1957, where he was normally talking to one of his colleagues that the molecules should flow from change their forms so that the genetic information is flowing, right? But then formally it was published in 1958 under the title On Protein Synthesis, where Francis Crick said that DNA molecule this DNA molecule will transcribe itself to form an RNA molecule. Now, this RNA molecule will again undergo another transformation where it is called as translation to get converted into a protein. Now, this protein will be functional inside any organism to carry out the particular function. Now, what happened later on in 1970s, two more scientists named David Baltimore and Howard Temin, they both proposed that no, DNA was able to replicate back to itself. The mRNA or RNA that is formed, it was able to get replicated back to the DNA structure. Now, this was because of an enzyme called reverse transcriptase. They saw this in one of the viruses, and again another new model of central dogma was proposed where they said that DNA will be able to get transcribed to RNA, but RNA will also be able to get transcribed back, that is reverse transcription, to form DNA. Now once RNA is formed, it will translate itself to a protein which will be functional. Now this protein is important and it will form the important molecules which is responsible for carrying out the different functions inside a body. Okay, so now since we know that the genetic information is flowing in this manner, now we got to know the importance of this DNA molecule to replicate itself so that it maintains its integrity and it is transferred from one generation to another. Now, let us understand what is the replication of DNA. Now, since we all know this is the structure of DNA, it was proposed by Francis Crick and Watson. This shows that it is made up of nitrogenous bases, which is adenine, thymine, cytosine, and guanine. Okay, now what happens? Replication. If we'll see replication in a very simple term, replication is nothing but making a copy of one molecule which is already existing. We can see here it is a biological process through which a cell makes an identical copy of its DNA. So the DNA, whatever bases are present here, the exact same copy will be made and the daughter cell will have the same basis whatever is present in the parent cell. Okay, and this is important because it helps the genetic information to flow from the parent cell to the daughter cell so that the genetic information is not lost and it is passed from one generation to another. Okay, now this DNA replication is very important because in day-to-day life our cells has to undergo a lot of work, and because of which we lose some of the cells, they are damaged, they are lost, they die, and hence we need it for growth, repair and reproductions, right? Okay, now coming to the types of replication. There are three major types of replication which was thought when Francis Crick proposed the structure of DNA. Now, the first type is the most important type, which is actual type, and this happens in DNA molecule. This is called as semi-conservative replication. Now, this was proposed by Michelson and Stahl in their experiment carried out in 1958. This we will know about later. Now, what happens in this replication? This is the parent strand, right? What happens? It will separate. Both the strands will separate themselves, and the daughter molecules will carry one strand from the parent and another strand will be newly synthesized. This means that, as you will see, this strand is the parent strand, right? So this will have the genetic information of the parent, and the newly synthesized strand will have the new information. This way the genetic information is not lost and it is transferred from one generation to another, right? As we can see here also, this is the parent strand, this is the newly synthesized strand. In the second generation, again when it is replicating, the parent strand will be here and the newly synthesized strand will be here. This will again form a template and a newly synthesized form will be here, right? So this is called as semi-conservative because only one strand is getting conserved, so it is called as semi-conservative type of replication. Next is called as conservative type of replication. Now conservative, the name itself suggests that the entire molecule is going to be conserved. Now this was again thought earlier when the DNA molecule was found, that it might be possible that the DNA molecule is replicating in a form where these two strands, they both separate from each other, right? This will separate like this, it will form two different strands, but this number one strand and number two strand, okay, this will undergo replication to form one of the daughter strand, and this will again undergo replication to form one of the daughter strand. Now what will happen after the replication is completed? These two will come together to form one molecule, and newly synthesized daughter molecules will come together to form the another molecule of daughter DNA. Then again this will undergo another round of replication in the same manner, hence producing different types of molecules. But this was again discarded when Michelson and Stahl performed their experiment to propose that no, it was the semi-conservative type of replication. Okay, now the next we have dispersive replication. Now dispersive replication again shows that this DNA molecule, which is the parent strand, it is going to separate itself in the earlier way as it was. It will separate itself into two strands like this. Okay, now these two strands will break itself into different fragments, right? These both the parent strands will break themselves into different fragments, and each fragment was supposed to undergo separate round of replication. Okay, so every daughter cell will undergo separate round of replication and they will form their own daughter strands. Okay, now when this entire replication is completed, what happens is all the newly synthesized strands along with the parent strand, they come and combine together to form this molecule. See, we can see two different molecules where both the parent strand and also the daughter strands are formed here and there, right, in the same molecule. And again in the next generation of replication the same thing happens, but this was also discarded by Michelson and Stahl, as it shows that it is not going to, as the DNA is not replicating in this manner. And this was also true because if the genetic material will undergo fragmentation, it has got a high chances of losing some of the genetic information which is very crucial for passing from one generation to another. And this way it was all two forms of replication were discarded and only the semi-conservative form of replication was accepted, that yes, DNA undergoes semi-conservative form of replication to replicate itself into a daughter strand. That's it for today. Thank you so much. Please subscribe our channel and follow along for more. Thank you so much.